Welcome to my presentation. I am Snigdha Bharadwaj, and today in the continuation of microencapsulation presentation, we are going to discuss the various type of mechanism of drug release as well as the evaluation parameter, right. So these are the mechanisms of drug release through microencapsulation encapsulated particles. So here various mechanisms have been proposed for drug release from microspheres, which are as follows. As I have told you that microencapsulated particles can be termed as microspheres, microbeads, micro particles. So it is all same if I say microencapsules or if I say microspheres, microbeads and whatever in micro range. So here are some mechanism of drug release through microspheres. First mechanism is degradation controlled monolithic system, diffusion controlled monolithic system, diffusion controlled reservoir system and erosion. Total four mechanisms have been mentioned for the drug release of active constituents through the microspheres. So we will discuss one by one in detail. Move on to next slide. Next slide. The first one is degradation controlled monolithic system. As you can see in the picture, the microsphere is the polymer coated encapsulating the core material. Core material represent the drug or active constituent here. So this is the pictorial representation of the microencapsule. The drug is encapsulated in within the polymer shell. So in degradation controlled monolithic system or the polymer matrix, so the degradation of matrix, polymer matrix has been, degradation of polymer matrix is occur during the release of the drug from the system. System is the microsphere. It renders the degradation of polymer matrix and it will gradually release the drug out, from within the polymer matrix to the outside environment. So this is how the one sort of mechanism is degradation of polymer monolithic system. Next, move on to the slide of diffusion controlled monolithic system. So basically drug is released or drug release occur by diffusion followed by the degradation of matrix. Again the first one is, as you can see the previous slide, it is degradation and next slide is diffusion. So both are the different terms, right. Diffusion means, yes, diffusion means passing the membrane and the degradation means degrading the polymer membrane, right, degrading the polymer membrane. So here it is occur if it is done by diffusion control monolithic. It depends on the property of polymer which we are using to coat the core material. Here you can see the drug is encapsulated in polymer shell and after this diffusion of drug, that means the drug particles come onto the surface of polymer membrane and diffuse out to the environment, outer environment, right. Here then you can see slow diffusion occurs during the release mechanism and finally the degradation will occur of the polymer membrane at the end of the duration, at the end of the system time. So initially the drug will be monitored through diffusion system and later the degradation will occur in which the polymer membrane totally or completely degrades and releases the remaining content of active component, right. Next, diffusion controlled reservoir system. In this you see the matrix form of the microsphere is there. For the reservoir type you see the double layer polymer membrane or the thick membrane of polymer you see. So the drug encapsulated in the reservoir type of microsphere formulation and now the drug is diffusing out through the reservoir slowly and after the times after the long time the polymer erodes means again the degradation, or it will burst and release the remaining content of active component, right. So, this is how the third, this may be a mechanism of drug release from the microspheres. Now, the fourth one and the last one is erosion based release. So, in this the polymer reservoir type, the due to pH and enzymatic activation. So, polymer membrane, the outer membrane of polymer may degrade or erode completely, and it will release the whole amount of drug from the microspheres, right. It is activated only in the case of pH enzymatic attack, if the drug is not compatible, and because when we administer the drug in the body, then all the enzymatic and body thinks that the drug is foreign material. So, all the enzymes and the pH will also play the important role in drug administration. So, this is one of the mechanism. Now, we will discuss about the evaluation parameters. When we make microcapsules or any kind of dosage form in the pharmaceuticals, so how we can assure that the drug is up to the mark or the formulation formed is up to the mark. So, we do generally testing of those preparations. So, as you know, in terms of tablet or capsules or solid oral dosage form, we can say that hardness, dissolution, disintegration, friability, drug content determination, weight variation, all these are the parameters through which we can evaluate the oral dosage form like tablets and capsules, that the drug is up to the mark or not, or formulation we make is up to the mark or not. So, similarly, in case of microspheres or microencapsulated particles, some of the parameters have been mentioned to ensure the quality of the dosage form prepared during the manufacturing process. Only after the evaluation we can assure that the preparation we have made or prepared is of standard one quality or not, right. So, here are some evaluation parameter which we are going to discuss in detail one by one. So, in this picture you can have a look that the particle size and shape, we are talking about the microspheres. Spheres means spherical shape particle, right, in micro range. So, micro particles shape and size plays an important role. So, this is why it is mentioned here. Percentage yield, the total amount of drug or and polymer and the ultimate finished product, how much we are getting, right. Drug content determination, in vitro release studies, entrapment efficiency, density determination, isoelectric point, angle of contact, electron spectroscopy for chemical analysis, and ATR, FTIR. So, these are some evaluation parameters, although apart from these there are variety of evaluation parameters which we can do as per the nature of the preparation of microsphere prepared. For example, magnetic microspheres. So, in case of magnetic microspheres we can have some different kind of evaluation parameters to check the magnetic properties of the microspheres present or not. So, it goes with the variety of or nature of the microspheres we prepare. So, if we talk about the generalized evaluation parameter, so these are the important parameters which we can do to ensure the quality and the nature of microsphere prepared. So, move on to next slide, we can have a single evaluation parameter in detail. Particle size and shape. Basically, particle size and shape can be evaluated or analyzed using high equipment, sophisticated equipments like light microscope, SEM, scanning electron microscopy, right, CLSM, confocal layer scanning microscope are widely used to analyze the size surface morphology of microcapsules. So, SEM, TEM, confocal layer, these are some laser microscopy. So, these are some high advanced technology to analyze the surface morphology and the size, shape of the particle to understand the basic sphericity of the particle prepared. How can we say that we can have variety of microparticles in the manufacturing process? Because I have told you that polynuclear, multinuclear, irregular cluster. So, we can confirm those type of particles or presence in the microsphere sample using these technologies. Although you can have an idea about the surface layer type, irregular or the smooth surface textures there on the polymer layer membrane coating membrane onto the core material. So, this is how we can analyze the morphology of the particles during the preparation. After the finished product we get, we will send the sample of properly packed sample to the analysis for the analysis, and using these equipments we can have the appropriate size. Yes, we say that the micro particle or microencapsulated particle should be lie within the range of 1 to 5000 micrometer. But how will you confirm this? Using these techniques you can confirm the size of single microsphere and the uniform size distribution. Say we can't say that single molecule or all the particles having same size. No, it is not at all possible. Yes, though some sort of similarity and uniformity you can achieve, but not 100%. So, this is how you can have how much particle range is present in the sample, and so that you can use it for further investigation. Now, the drug content determination. Drug content determination basically you can check the drug content in the sample. Yes, if you use 100 mg of any core drug directly to make formulations like tablet, capsules, then you can expect plus minus 5% the percentage variation in the tablet. But when you use the drug with polymer to make microencapsules, then you will have to check the content of drug present in the microspheres, because you use the solutions like you use several mixtures of aqueous phase, organic phase, then how will you identify the drug content? So, it is also important, yes, because during manufacturing process when you use two different kind of mixtures to make the solid particles, then you will filter out those solid particles from to the mixture, and that will be wasted, right. So, only the concern here is how much quantity of drug is utilized within the system of microsphere preparation. So, this is an important concern here. So, drug content determination, how we can check it? It is a generalized procedure. It may vary type to type depending on the type and nature of the drug you use. So, you can have variety of procedures representing drug content determination. If you talk about the oral delivery of microsphere, then you will use the gastric fluid or lumen condition things, right. So, in that case you will use the media of pH 1.2 representing the gastric fluid. And if you use the topical topical application microsphere, then the pH will get like 6, 6.6, 6.8 phosphate buffer. The media will get changed every time as per your route of administration. So, in drug content determination, equivalent sample of microcapsules what you have prepared in your lab is dissolved in the suitable buffer. As I have told you, if you are making it for gastric delivery, then you will take the gastric fluid, otherwise you will take the phosphate buffer of pH varying pH based on your route of administration. So, dissolved in the suitable buffer to make standard solution and then filtered, because it is not only containing a drug, it is also containing a polymer coating. So, we do not want the polymer to be in the sample of testing for analyzed drug content determination, we only want the drug present in the microsphere or how much quantity of drug encapsulated in the microsphere. So, the solution is get filtered, and now the further sub dilutions, after making standard solution, the further sub dilutions can be made to check the different types of concentration present in different samples through UV analysis. So, UV spectrophotometric analysis can be used to identify drug content at basic level, although variety of techniques are also there, like you can identify the content using HPLC, HPTLC. So, at basic level you can have the concentration through UV analysis. Percentage yield is also important. If you are using one drug and polymer ratio in 1:1, so you can expect 200 mg total in amount, but you are getting only 50 mg microsphere out of that 1:1 ratio. So, the yield is very low, right. So, you need to identify which type of procedure is giving you the highest yield in terms of weight. So, the procedure should be productive, so that you can compare. Yes, you have variety of method of preparation for microspheres, right. So, you can then choose that this method is giving. So, every method is giving different kind of specifications, like spray drying will give you the very fine small size particle, whereas coazerbation will give you matrix type, and air suspension method will give you the higher size particle. So, according to your specification you can choose your method of preparation. So, this is how we can calculate percentage yield, how much quantity we have taken during the manufacturing process of drug as well as polymer, and how much quantity we have got after the manufacturing process. So, this is how we calculate percentage yield. Now, first important entrapment efficiency and loading. As I told you that entrapment efficiency when it is reducing and when it is increasing in my previous lectures. So, in continuation of that only, how we can calculate entrapment efficiency, how we can say that entrapment efficiency of the microsphere is low or high. Entrapment means encapsulation of the core particle. The polymer is able to entrap the core particle well or not. We can identify using this technique. So, percentage entrapment efficiency can be determined by allowing the washed microsphere to lysis, followed by UV visible spectrophotometric analysis. Yes, first you will take this sample and of microsphere what you have prepared in your lab or after manufacturing to check entrapment efficiency, and then you will use some sort of lysing agent like Tritonex and many more said, right. So, many more you can use, and then after lysis of microsphere you can filter it out, the solution you will get, you will make further subdilution, and then you can calculate the percentage EE using this formula, actual content by theoretical content divided by 100, as you are taking percentage. So, actual content means how much content you have received through UV analysis, right, and theoretical, say you use 100 mg of drug in the system. Initially you have added 100 mg of any drug of which you are making microsphere initially in the system during manufacturing. So, this is how you can calculate entrapment efficiency. Theoretical yield, you will get the percentage of drug entrapped within the polymer system. So, next is your density determination. Accurately weighed sample is placed in cup of multi-volume pycnometer. Helium is introduced at constant pressure in chamber, right. So, this is how we can identify density. The density basically represent the weight or the size of the particle. So, this is one of the important parameters to evaluate microcapsules in which the helium is introduced at constant temperature and we use pycnometer to check the density. Okay. Helium is introduced at constant pressure in chamber and is allowed to expand. Two consecutive reading of reduction in pressure at different initial pressure are noted to determine the density of microcapsule. This is how you determine the density using pycnometer. Next, your attenuated total reflectance Fourier transform infrared spectroscopy. It is also said ATR, FTIR. FTIR, as you know, we can use this technique to identify or to interaction between drug and polymer. So, you can also use this technique to see whether the drug is adversely interacting with the polymer or polymer is adversely interacting with the drug encapsulated within. Otherwise, if you do not observe these kind of interaction, you will not be able to understand the drug release profile. If drug release is affected due to this interaction, negative interaction between drug and polymer, then you may not have adequate release of the drug, right. So, this is also important concern. FTIR is used to determine the degradation of the polymer matrix of the carrier system. Another aspects of this ATR FTIR is to determine the degradation matrix of the polymer carrier system, like carrier is here is microsphere. So, surface of the microsphere is a measure by ATR, right. So, this is the equipment through which you can evaluate the surface of the microspheres, and both gives the information about the surface composition of the microsphere, as the polymer coating is the outer coating. So, you are getting the information of polymer surface only, right. Microsphere depending on the manufacturing procedure and conditions. So, this is how you can measure the your surface environment and the surface characteristics using ATR FTIR. Electron spectroscopy. Next is your electron spectroscopy. Surface chemistry of microcapsule can be determined using ES, electron spectroscopy, for chemical analysis, atomic composition of surface, right. What are the composition, what are the functional groups attached are responsible for making bonds between drug candidate and the polymer, what are the interaction points through which drug is making bond with the polymer to encapsulate within the polymer shell. So, these information can be revealed using these electron microscopy spectroscopy, as these are giving you the information regarding your atomic level, regarding atomic structure and atomic groups compositions. Now, angle of contact, it is very important, wettability and swelling index. We can have these ideas using this angle of contact parameter used to determine wetting property. If it is not at all hydrophilic water adaptable, then wetting will get 0. That means, the microsphere will not get dissolved in the water environment. So, these are the considerations that only the polymer is totally water insoluble or some wettability is there, then it is soluble in water or aqueous phase as well. So, it is used to determine wetting property of the microcapsule, that is hydrophilic or hydrophobic nature, and of angle of contact is measured at solid air water interface. So, using these parameter, you can have an idea about the polymer hydrophilic and hydrophobic nature. Understand? Next is isoelectric point. So, isoelectric point, basically microelectrophoresis apparatus is used to measure the isoelectric point, that measures the electrophoretic mobility of microcapsule, from which isoelectric point can be determined, right. The velocity at different pH measures particle movement in time over distance of 1 nm. These are some specification of the equipment. By using all these specification, you can have isoelectric point of this particular type of microsphere you are analyzing. In vitro release study, it is very important, because in case of tablet and capsules you determine dissolution studies, and now for microsphere the term is now in vitro release. It is all similar, but the procedure is slightly different. The test is carried out using rotating paddle apparatus as you use for tablet and capsules, and the RPM revolution per minute speed will be like 100, and paddle type apparatus will be used. It is basically not specific, it is not specific that you use only paddle type. Based on type and nature of your formulation, you can change your apparatus. So, the dialysis is another method used to study release of drug from microsphere. Either you can use the dissolution apparatus paddle type, or you can use dialysis as another method. Microcapsules are kept in a dialysing bag or tube membrane while a dialysing media is continuously stirred. So, you can make the in vitro in vivo correlation that you are providing. Why you are analyzing in vitro release? Because you want to make some in vivo in vitro correlation between your that you are measuring this release, and you are getting that it is the amount in which is releasing in this time period. Then you can correlate that in body also it will be like same. If you are not comparing the in vitro and in vivo correlation, then you cannot make a point that the release will be same in within the body or not. For if you are using microsphere for skin purpose or topical delivery, then you will use this dialysis technique, or if you see, so that you can see the permeation and the penetration pattern of the drug, how much quantity is penetrating through the skin or across the skin. So, this is how you calculate in vitro release. Like amount of drug release can be calculated by taking concentration, volume of dissolution medium, dilution factor, and dilution factor means you are taking some specified amount of media, like say 900 ml if we talk about dissolution apparatus, and when you take sample of 5 ml after specified interval, you are maintaining sink condition or not, means you are adding up the fresh blank solution to the 900 ml flask or not. If you are not adding, then you have to put the dilution factors at every interval, that if you in 900 ml when you take 5 ml, then it will become 895 ml, right. So, accordingly the dilution factor will be calculated. Percentage drug release, the amount of drug release divided by strength into 100, as amount of drug release can be calculated using this formula, and overall percentage drug release can be calculated uses this amount of drug release you will get from this, and the strength percentage drug release from this. There are some marketed products you can see that already coming in the market as a commercial products, Respideron as Constan, Respideral, and companies Janssen Alkermes, and these are variety of marketed preparations of many more preparations are also there, but I have listed some few microsphere preparation coming for the market. So, these are the main ingredient using the microsphere, in which these are encapsulated in the microsphere and can be used for variety of purposes. So, here is how you understand all the concepts related to microspheres, that the basic introduction, the method of preparation, and the mechanism of drug release, as well as the evaluation parameter. I hope the information I gave you will be fruitful, and you will get benefited by this, and thank you for listening. Take care, stay safe.